I'm Máté Manczinger. I'm leading the Systems Immunology Research Group in Szeged in the BRC, and I'm a junior mentor for 6 years and also a mentor for 2 years in this wonderful program. And I'm happy that I can introduce my main research focus in this lecture. Okay, I'm trying to. Okay. So we are focusing on immune recognition of several substances and pathogens. So the immune system's main role is to differentiate between the harmful agents and the non-harmful agents. And in harmful agents, we can find cancer, for which the immune system is elemental to destroy and prevent the formation of cancer cells. And the basis of anti-tumor immunity is the presentation of mutated, modified peptides on the surface of the cancer cell. And this presentation is carried out by the so-called HLA human leukocyte antigen molecules. And if T cells of the immune system can recognize this complex of HLA molecule and the modified peptide, then the immune system has the possibility to destroy the cancer cell. So we can say that this is the prerequisite of tumor destruction. However, the cancer cells has developed strategies to avoid immune destruction. For example, they can express molecules, for example, the PD ligand 1, with which they can inhibit the activation of T cells. And fortunately, recently, around 2 decades ago, drug has been developed with which this inhibition can be blocked and the T cells can be reactivated. We call this the cancer immunotherapy or immune checkpoint blockade immunotherapy, and I can say that it has revolutionized the oncotherapy. Because those patients who had previously very poor survival had much better survival nowadays, and even late-stage patients have the chance to survive or even cure from metastatic disorders. However, yes, and the 2 researchers having the main role in the development in these drugs was awarded a Nobel Prize in 2018. But we have these patients here who do not respond to the immunotherapy. And it's very important to find out why they do not respond. What are the factors that drive the anti-tumor immune response against the cancer cells? And until recently, a quantitative approach governed. It suggested that the more mutations there are in the cancer cells, it is more likely that these mutated peptides will be developed, and consequently, the anti-tumor immune response will be more effective, and the patients will respond to the immunotherapy more likely. However, it has been changed in the previous several years, and in the first case, I would like to show our example why quality is more important sometimes than quantity. We focused on these HLA molecules that bind these modified peptides, and they are essential for the development of an anti-tumor immune response. They are highly variable, and consequently, it's very unlikely that in this room there are 2 individuals carrying exactly the same HLA molecules. And different HLA molecules have different binding specificities, so they are specific for different peptides. And they have a very interesting feature, the peptide binding repertoire size. It means that certain variants are able to bind only a low number of different peptides, but other variants bind a much higher number of these peptides. And the former ones are called specialists, and the later ones are called generalist molecules. And one would think that carrying generalists is more advantageous because it's more likely that mutated cancer peptides will be on the surface of cancer cells. However, we checked this and found exactly the opposite. We found that those individuals who carry generalist molecules, so they have a high or a large peptide binding repertoire size, they survive much worse than the others. And we asked the question that why? Why can it be? We had many hypotheses. And I will show you the most likely one. Because we found that with increasing repertoire size, the immune system will be unable to differentiate between the original peptides and the mutated peptides. This is because these mutated peptides are usually different from the original ones in only one amino acid. And generalist molecules are not so specific for anything. They also present to the immune system the original non-mutated peptide, and consequently, the immune system cannot decide which one is the mutated one and which one is the original one. At the same time, the specialist ones only present the mutated peptides, so the immune system has a much easier task. And according to this, we found that in samples with generalist HLA molecules, there is tolerance, so the immune system tolerates the tumor. It thinks that it's ourself, and consequently, the patients will not respond to immunotherapy, and the survival will be very poor. However, we can ask the question, is the presentation of a high number and a high quality of mutated peptides is enough for good survival? And I would like to briefly introduce you our current research focus, which suggests that it's still not enough. HLA molecules have two classes. Basically, the research, and we also focused on HLA class 1 molecules, but it has been recently suggested that HLA class 2 molecules are also very important for immune recognition. I don't want to go into details because time limitation, but we focused on both classes. And we found that there are combinations that are associated with very good survival of cancer immunotherapy patients, and there are combinations that are associated with very bad survival. And we try to examine what could be the explanation for that. If we consider the quantitative approach, we would think that the HLA 1 and 2 molecules in this combination present a higher number of mutated peptides. We found that both the beneficial combinations and the detrimental combinations presented a very high number of mutated peptides in melanoma. And additionally, we found that the detrimental combinations were the one, I tried to target it, so the detrimental combinations were the ones that presented highly immunogenic mutations. So presented those mutations that can be easily recognized by the immune system. So again, we were a little shocked, and we tried to find out what can be the explanation. And we found that in early tumor stages, those individuals that carrying detrimental combinations in the tumors, in primary tumors, in early tumors, we can see a cytotoxic immune response. And it forces a very strong immune-mediated selection on the tumor. And consequently, in metastasis, the HLA molecules will be downregulated, and the neopeptides, these mutated peptides, will be lost on the genetic level or on the transcriptomic level. They will be downregulated. So there will be an immune evasion, which is not reversible by cancer immunotherapy. At the same time, in beneficial combinations, in primary tumors, there is no active immune response, but in metastasis, there is an active and effective cytotoxic immune response. So we can say that the timing here is perfect for the anti-tumor immunotherapy. So as a conclusion, we can say that quantity is not enough, and quantity and quality together is also not always enough for an effective anti-tumor immunity response. There is a golden mean, and we have to consider a lot of factors. So patients should be treated in a highly personalized manner. For example, if somebody carries detrimental combinations, these patients cannot take or have cancer immunotherapy in their metastatic stage because it's useless. They will not respond to the therapy. But during the primary stage, they are very likely to respond to these therapies. So I would like to thank everybody who helped in this project, and thank you for your attention.
Thank you very much for this very exciting and very clear presentation about the perspective of immunotherapy in certain cancer stages. And if you have any question or remark, just come forward, and I encourage you to ask Dr. Manczinger. Is there anybody who... And the type of the tumor, you see some difference? You mentioned at the early stage and the metastatic stage, but what type of cancers you can talk about here?
Thank you for the question. So basically, we focused on melanoma in the second project, and we focused on melanoma because this is one of the most immunogenic tumors. It carries the highest number of mutations. These phenomena could be available also in non-small cell lung cancer, which is another type of immunogenic tumor. But these two types on which we focus, we want to widen our perspective further, and these are our plans currently.
Thank you very much. In the meantime, there are some other questions. Please, go ahead.
As I understood of the presentation, when you want to mark the mutated protein with a more general and not so specific molecule, what you built, you said that the immune system will be more tolerated to all the presented cells, so it won't work. And why doesn't the immune system kill all the presented cells? Why does it become tolerant?
Yes, so the mechanism, I didn't want to go into detail, but the mechanism behind this is that HLA molecules usually present not only mutated peptides, but they present some peptides also from our own cells. So basically, it's true constantly. And so in the specialist molecules, as they have a narrower binding repertoire, this is unlikely that these molecules peptide the original counterpart of the mutated peptide. And the generalists are very likely to present the original counterpart. And unfortunately, or fortunately, I don't know, T cells are cross-reactive. It means that the same T cell is highly specific for the original and the mutated peptide as well. And if the original is presented by the HLA molecule, then a tolerance is developed in the immune system against that peptide, and as cross-reaction is present against the mutated peptide too. So this is the mechanism.
Thank you very much. There is time for one more question, if there is any in the room. Oh, please. Sorry.
I'm sorry.
Out of my vision.
Well, just a small point. If I understood you correctly, time is a very important parameter for immunogenic therapy. And I have a translational question, if I may say so. Cancer presents many times with its metastasis, and so the primary stage might be missed easily. Does it have any implication for the potential outcome for immunotherapy, or how should we interpret your findings in this, from this viewpoint?
Yes, absolutely. So, especially melanoma, we are quite lucky because it's visible and we can easily diagnose it in early stages. But in many cancer types, there are also already metastases. And the clinical consequence of this phenomenon that although immune checkpoint inhibitor can be effective in the metastatic stage, those individuals who carry these detrimental combinations should not take at least this immune checkpoint blockade immunotherapy, because they will not respond. They should be included in clinical studies or some studies with adoptive T-cell transfer therapy and other kind of therapies, which can or could have a much bigger effect. So yes, they gives us an option. So this biomarker can give us an option to include a patient in a study with which the patient has a risk to survive or the chance to survive.
Thank you very much. And now on behalf of the National Scientist Academy, we would like to recognize your excellent contribution toward the success of this meeting and to be a very effective mentor in the program. So please come forward.